Welcome to the deep dive. We're tackling a fascinating topic today, the world of nutrigenomics.
Yeah, it's definitely a hot area.
A listener sent in a really comprehensive research packet, and as always, our mission is to, you know, extract the most important, surprising, and actionable insights for you.
Exactly. We've sifted through quite a bit of science here to bring you the key takeaways, saving you, well, hours of reading.
Our goal today is really to help you move beyond that generic dietary advice and understand how your own unique biology interacts with the food you eat. We're aiming for those aha moments, without getting totally bogged down in overwhelming
It's
details.
easy to get lost in the weeds with this stuff.
For sure. And to guide us on this journey into the molecular world of food, I'm delighted to have our expert with us.
Thanks. Happy to be here. I'm really eager to get into this one.
So let's just jump right in then. For so long, we've been given these broad, sweeping dietary recommendations, haven't we?
One size fits all.
Exactly. But what's becoming increasingly clear is that we all respond to food in really individual ways. And that's where nutrigenomics and its close relative nutrigenetics come into play.
Right.
It's about understanding that unique biological conversation that happens, between the food we eat and our bodies at a really fundamental level. It feels groundbreaking.
It really is. And just maybe to clarify for everyone, nutrigenomics looks at how the nutrients and compounds in food can actually influence our gene expression, basically how they tell our genes to be more or less active.
Okay.
Nutrigenetics, on the other hand, looks at how the genetic variations we're born with affect how we respond to those nutrients.
Gotcha.
They really work together,
Yeah.
to paint a picture of truly personalized nutrition.
Right. So it's not just about calories or macros in the simple sense. It's about the biological information within that food and how it, talks to our genes. That's quite a powerful idea.
Absolutely. And cornerstone of the nutrigenetic side of things is the concept of single nucleotide polymorphisms, or SNPs, as they're usually called.
Yeah. SMP is right. Heard that term.
Yeah. These are simply points in our DNA where there's a difference in a single building block. Think of it as a tiny individual variation in our genetic code at one specific spot.
Like a tiny typo, almost. Or maybe more like a different spelling variation.
Yeah, maybe more like a spelling variation. And the amazing thing is we each have millions of these SNPs. They're incredibly common. They're a huge part of what makes each of us unique.
Okay.
And importantly, these are usually just normal variations, not necessarily defects or problems with our genes, just differences.
Okay, so these tiny differences, these SNPs, how do they actually influence, say, how we respond to food or maybe our likelihood of developing certain health issues?
Well, depending on where a SMP occurs in the DNA, it can subtly alter the way our bodies make proteins.
Okay.
Now, these proteins might be enzymes that speed up reactions,
Mm-hmm.
or maybe they're involved in how our cells communicate. SMP can slightly change how well these proteins work, maybe make them a bit faster, a bit slower, a bit less stable. And that, in turn, can affect how we process nutrients, how our bodies handle inflammation, or how we respond to different foods. It's a very delicate system.
So even a seemingly tiny change in our DNA can have this kind of knock-on effect on how our body interacts with what we eat.
Precisely.
That really highlights why what works brilliantly for one person might just not work the same way for someone else, right?
Exactly. And this is the core principle, really.
Yeah.
Our individual genetic makeup often trumps those general dietary rules we've all heard.
Right.
Population-wide guidelines are a good starting point, sure, but they can't possibly be optimal for everyone because they don't account for this underlying biological individuality.
Yeah.
It also kind of validates those times you might have felt, this healthy food just doesn't seem to agree with me. There could very well be a genetic reason behind that feeling.
Okay, this is fascinating. Let's dig a bit deeper then. Our genes are the initial blueprint, as you said, but it sounds like that blueprint isn't totally fixed in terms of how it's expressed.
Yeah.
That's where epigenetics comes in, yeah.
Exactly right.
How our environment, and especially our diet, can influence how our genes actually behave day to day.
Precisely. Think of epigenetics as like a control panel that sits on top of our DNA sequence. It involves modifications to the DNA or to the proteins wrapped around it
Okay.
that change how active our genes are. But crucially, without changing the underlying A's, T's, C's, and G's, the code itself.
So if the DNA is the instruction manual, epigenetics is kind of like the highlighter, pens, and sticky notes that tell the cell which instructions are important right now and maybe how loudly to read them.
That's a fantastic analogy, yes. And what's really exciting and frankly encouraging about this field is that many of these epigenetic marks are reversible.
Ah.
Unlike our fixed DNA sequence, we actually have some power, some agency to influence the activity patterns of our genes through what we eat and how we live.
That's incredibly empowering. It's not all predetermined. So how does our diet actually, flip these epigenetic switches? What are the main ways this happens?
There are several key mechanisms. One major one is called DNA methylation. This involves adding a small chemical tag, it's called a methyl group, directly onto the DNA molecule itself. This often acts like a dimmer switch, usually turning gene activity down or even off.
Right.
And certain nutrients, think methionine, folate, B12, choline, are absolutely essential because they provide the raw materials, the methyl groups, needed for this process.
Ah, the methyl donors.
Exactly, the methyl donors. And interestingly, in conditions like type 2 diabetes, scientists have observed specific changes in DNA methylation patterns in genes related to insulin and glucose metabolism, strongly suggesting a link to diet and lifestyle.
So these nutrients are literally providing the tools to adjust our gene activity levels.
Wow, okay, what's another way?
Another important way is through histone modification. You see, our DNA isn't just floating around, it's wrapped around proteins called histones.
Right, like thread on spools.
Exactly like thread on spools. And this whole structure is called chromatin. Now, chemical modifications can be made to the tails of these histone proteins, and this changes how tightly the DNA is wound.
If it's wound really tightly, genes are harder for the cell machinery to access and read, so they tend to be less active or silenced. If it's looser, the genes are more accessible and can be turned on more easily.
But the packaging matters.
The packaging matters a lot, and different dietary components can influence this. For example, some studies suggest saturated fats might increase the activity of enzymes that tend to loosen the chromatin, while certain polyphenols, those beneficial compounds in colorful plants, can influence enzymes that help keep it more tightly wound, where appropriate, potentially silencing genes we don't want active.
So what we eat can influence the physical structure, the accessibility of our DNA. That's amazing. And you mentioned a third way.
Yes, a third key mechanism involves small molecules called non-coding RNAs, particularly a type called microRNAs or miRNAs.
MicroRNA, okay.
These are tiny RNA molecules that don't code for proteins themselves, but they can bind to messenger RNA, that's the molecule that carries the instructions from DNA to the protein-making machinery. When a microRNA binds to a messenger RNA, it can either cause it to be degraded, basically chewed up, or it can just block it from being translated into a protein. Either way, it effectively silences the gene's message.
And diet influences these too?
You bet. Dietary compounds like curcumin from turmeric, resveratrol from grapes, quercetin from onions, these have all been shown in research to influence the levels and activity of these microRNAs.
Wow, okay, so it's like a three-pronged attack. Diet can influence DNA methylation, the histone packaging, and even these RNA regulators. That really highlights the profound influence food has at a molecular level.
It really does. And when we look at specific dietary components, the connections become even clearer. We mentioned methyl donors, folate, B12, methionine, choline. They're crucial for producing a molecule called SAM, or SAM.
Same, yeah.
Which is the body's main methyl donor. If we don't get enough of these foundational nutrients, it can lead to problems with DNA methylation, potentially increasing our risk for certain diseases over time.
Makes total sense. What about those colorful plant compounds we always hear are so good for us? The polyphenols.
Right, polyphenols found in berries, tea, coffee, dark chocolate, spices, you name it, they're fascinating epigenetically. Some can actually inhibit the enzymes that add methyl groups to DNA, DNMTs, or the ones that remove those activating tags from histones, HDACs.
Okay, so they stop the silencing.
In some cases, yes. This can potentially have the effect of turning back on genes that might have been inappropriately silenced, like tumor suppressor genes, for example. So these compounds can help our bodies maintain a healthy, balanced pattern of gene activity.
That's incredible. So it's not just antioxidants, they're actually talking to our gene expression machinery. What about something like sulforaphane from broccoli? I hear about that one a lot.
Sulforaphane is another great example. It acts as an HDAC inhibitor, meaning it helps keep genes on by preventing the removal of those activating acetyl tags from histones. It's been shown to have epigenetic effects that can help suppress certain cancer-related genes. And we're constantly learning more about how different types of fats, like omega-3s versus saturated fats, and even vitamins like D and C and minerals like selenium and zinc, all participate in this intricate epigenetic conversation.
So it's really not just the basic vitamins and minerals, but these specific bioactive compounds within whole foods that are having these quite significant effects.
Exactly.
And of course, it's not just about diet, right? our overall lifestyle must play a role too.
Oh, absolutely. Chronic stress, for instance, has been shown to alter epigenetic patterns. Physical activity, on the other hand, generally has beneficial impacts on DNA methylation and histone modifications.
Okay.
Then there are environmental exposures, things like air pollutants, toxins, even certain medications. Smoking is a classic example. It causes widespread changes in DNA methylation, though thankfully many of those changes seem to be reversible if someone quits.
That's good to know.
And even body weight itself, like obesity, is linked to altered DNA methylation patterns in genes involved in fat cell development, inflammation, and insulin signaling.
Right. So it really drives home that point.
Mm.
Our genes are more like a starting point, maybe a predisposition, but definitely not a fixed destiny.
Exactly.
It's this constant dynamic interaction between our inherent genetic code and how we live our lives, especially what we put in our mouths. This really ties into our previous discussions about how things like histamine intolerance or even ultra-processed foods have real, tangible consequences right down at the molecular level.
No.
But it also offers a huge sense of hope and agency, doesn't it? We seem to have more influence than we might think.
Precisely. Our genes might, load the gun, as the saying goes.
Yeah.
But our diet and lifestyle factors often pull the trigger. Understanding this interplay is really key to feeling empowered about our health choices.
Okay, so we get the big picture. Individual genetics matter, and epigenetics means our choices influence gene expression. Now, let's focus on some specific genes that are really key players in this whole nutrigenomics landscape.
Right.
Let's start with MTHFR. It feels like everyone's heard about that one.
Yes, MTHFR. It stands for methylene tetrahydrofolate reductase. Quite a mouthful.
Definitely.
It's a really important gene involved in how our bodies process folate, which is a crucial B vitamin. The enzyme made by this gene is essential for converting folate from food or folic acid from supplements into its active form, called 5-MTHF.
Okay, the active form. Why is that important?
Well, this active folate, 5-MTHF, is needed for a critical process called methylation. And methylation is involved in just so many vital functions: DNA synthesis and repair, turning genes on and off, producing neurotransmitters like serotonin and dopamine in our brain, and even detoxification pathways like making glutathione, our master antioxidant.
Wow, so it has really far-reaching effects, much more than just folate processing.
Absolutely.
Now, what are the common variations, the SMPs, that people often talk about with MTHFR?
The two most studied SMPs are C677T and A1298C. If someone has the T version of the C677T SNP, especially if they inherit two copies of it, what we call homozygous TT, the MTHFR enzyme they produce tends to be less stable and doesn't work quite as efficiently, especially under certain conditions like low riboflavin status.
Okay.
The A1298C variant generally has a more subtle impact on the enzyme's function on its own, but can compound effects if present with C677T. And these variations are actually quite common, though prevalence varies across different populations.
So what are some of the real-world implications then? If someone has these variations, what might they experience or need to be mindful of?
Well, if MTHFR isn't working optimally, it can lead to lower levels of that active folate, 5-MTHF, circulating in the body.
Right.
This can indirectly affect how we use vitamin B12, too, sometimes leading to something called a methylfolate trap. One common consequence linked to less efficient MTHFR function, particularly the C677T variant, is elevated levels of an amino acid called homocysteine in the blood.
Ah, homocysteine.
Yeah.
I know that's linked to heart health risks.
It's exactly. High homocysteine is a known risk factor for cardiovascular disease. And because MTHFR is so central to methylation, reduced activity can also impact the production of those important neurotransmitters, potentially affecting mood. And it can impair the body's detoxification capacity by affecting glutathione production.
Okay.
There's also been research linking certain MTHFR variations to things like recurrent pregnancy loss and neural tube defects, although the picture there is complex, especially now with widespread folic acid fortification in foods. And some studies suggest links to anemia risk as well.
And you mentioned earlier a possible connection, an indirect one, to histamine intolerance.
Yes, indirectly. There's an enzyme that breaks down histamine inside our cells called HNMT. This enzyme relies on that same methylation process. It needs SAME, the universal methyl donor, to work.
Which MTHFR helps produce.
Exactly. So if MTHFR function is reduced, leading to potentially lower SAME production, then HNMT might not work as efficiently either. This could contribute to histamine building up and causing symptoms of histamine intolerance in some individuals. It's all interconnected.
That's fascinating how these seemingly separate pathways are actually linked biochemically. So if someone knows they have these MTHFR variants, What sort of dietary or maybe supplement considerations might be helpful?
Well, focusing on getting plenty of folate-rich foods, leafy greens, legumes is always a good foundation. For those with significant MTHFR impairment confirmed through testing and symptoms, supplementing with the active form of folate, 5-MTHF, might be considered, rather than folic acid.
Okay, bypassing the MTHFR enzyme step.
Exactly. Ensuring adequate intake of vitamin B12, often in the methylcobalamin form, and vitamin B6, perhaps as P5P, is also important as they work together in these pathways. Riboflavin, vitamin B2, is a key cofactor for the MTHFR enzyme. So ensuring good intake can be particularly helpful for those with the C677T TT genotype.
Right.
And things like choline are also important for methylation support. Monitoring homocysteine levels can be useful too. But it's really nuanced, especially the folic acid versus 5-MTHF debate. And it's best discussed with a healthcare professional knowledgeable in this area.
Definitely sounds like something needing professional guidance. Okay, let's tackle another big one, APOE, and specifically the APOE4 variant. We hear so much about its link to Alzheimer's risk. What's the primary job of the APOE gene?
APOE produces a protein, apolipoprotein E, that's essentially a transport vehicle for fats or lipids, cholesterol included, throughout the body, and very importantly, within the brain.
Uh-huh.
In the brain, it plays a critical role in delivering these lipids to neurons, which need them for maintaining their structure, for synaptic plasticity, basically learning and memory, and for repair processes. Think of it as the brain's delivery service for essential fatty building blocks.
Got it, a fat transporter. And what are the different versions or alleles of this gene that we hear about, the A2, A3, 4?
That's right. Those are the three main versions, or alleles.
Mm.
The A2 allele is the most common and is generally considered sort of the baseline or neutral version in terms of disease risk. The A2 allele is less common and is often associated with lower levels of LDL, the bad cholesterol, and might even offer some protection against late-onset Alzheimer's disease.
Interesting.
However, it's the APOE allele that gets the most attention. Carrying one or especially two copies of APOE4 is the most significant genetic risk factor for developing late-onset Alzheimer's disease. It also increases the risk for cardiovascular disease.
Wow. What is it about the APOE4 version that makes it have such a significant impact, particularly on brain health?
Well, the APOE4 protein seems to be less efficient at transporting lipids, particularly cholesterol, within the brain compared to the APOE3 or APOE2 versions. It's also associated with a more pro-inflammatory state, both systemically and within the brain itself, specifically involving microglia, the brain's immune cells.
Inflammation, okay.
In the context of Alzheimer's disease, APOE4 appears to accelerate the aggregation or clumping of amyloid beta protein into plaques, which are a key pathological feature. It might also impair the brain's ability to clear these amyloid beta proteins away effectively, and it seems to disrupt normal neuronal processes like endocytosis.
And you mentioned something earlier about iron.
Yes, that's a newer area of research.
Yeah.
Some studies suggest APOE4 might be linked to disruptions in iron regulation within brain cells, particularly at the blood-brain barrier, which could contribute to dysfunction. It's complex, but potentially another piece of the puzzle.
It certainly sounds complex. So for someone who knows they carry one or two copies of the APOE4 allele, what kind of dietary strategies might be particularly beneficial or protective?
Generally, the advice leans towards limiting intake of saturated fats, often found in red meat, full-fat dairy, and tropical oils like coconut oil.
Okay.
Conversely, increasing intake of unsaturated fats, especially omega-3 fatty acids found in fatty fish like salmon, as well as walnuts and flax seeds, is often recommended due to their anti-inflammatory properties.
Makes sense.
Emphasizing foods rich in antioxidants, think brightly colored fruits and vegetables, is also crucial to combat oxidative stress. And ensuring adequate intake of choline found in things like egg yolks and liver might be important for brain cell membrane health.
So dietary patterns like the Mediterranean diet often come up.
Yes, the Mediterranean diet, with its focus on olive oil, fish, vegetables, fruits, and nuts, aligns well with these recommendations. Some modified paleo approaches that emphasize whole foods, but are careful about the type and amount of fat, might also be suitable.
What about high-fat diets like keto?
That's where caution is generally advised for APOE4 carriers. While some might do okay, many individuals with APOE4 experience significant increases in LDL cholesterol on very high-fat ketogenic diets, particularly those high in saturated fat. Limiting refined carbohydrates, sugar, and alcohol is also usually recommended. It really comes down to being vigilant about fat quality and quantity. There was even a UK study suggesting lamb fat might be particularly problematic for ApoE4 carriers' cholesterol levels.
Really interesting. Okay, let's move on to FTO. This one is often dubbed the obesity gene, right? What's its main function?
Right, FTO, which stands for fat mass and obesity-associated protein, was actually the first gene strongly linked to common obesity risk through large genetic studies. The A allele of a specific SMP, RS9939609, is the one associated with increased risk.
Okay. And what does the FTO protein actually do?
Its precise functions are still being unraveled, but we know it acts as an enzyme, specifically an M6A demethylase. This means it removes a specific chemical tag from RNA molecules. This modification, M6A, can influence RNA stability, translation, and splicing, thereby affecting the expression of many other genes involved in processes like adipogenesis, fat cell development, and potentially even tumorigenesis.
Okay, so it's involved in regulating other genes. How does it seem to influence our weight or energy balance? Is it more about calories burned or calories consumed?
The evidence strongly points towards its impact being primarily on the energy intake side of the equation, influencing appetite regulation. People carrying the FTO risk allele, often report increased feelings of hunger, a greater preference for energy-dense, high-fat, and sugary foods, and potentially impaired satiety, meaning they don't feel as full after eating.
So it might make resisting those foods harder?
Potentially, yes.
Oh.
Interestingly, while some early mouse studies where the FTO gene was knocked out suggested effects on energy expenditure, most human studies haven't found a strong, consistent link between FTO variants and how many calories people burn at rest or during activity. So it seems more about driving intake.
Got it. Intake, not expenditure. And are there links to other health conditions besides obesity?
Yes, the FTO risk allele has also been linked to an increased risk of developing type 2 diabetes, cardiovascular disease, and potentially some types of cancer, although these links are often mediated, at least in part, through its influence on BMI and body weight. The research provided didn't show a strong direct link to iron metabolism, just to clarify that point.
Okay. Now here's a crucial question. If someone knows they have this FTO risk allele, are they just destined to struggle with their weight?
Absolutely not. This is a fantastic example of a gene-environment interaction. While the gene might increase susceptibility, lifestyle factors play a massive role in determining the outcome.
Like what?
Two key things stand out from research. First, protein intake. Some studies suggest that for individuals with the FTO risk allele who are trying to lose weight, consuming a higher proportion of calories from protein might be particularly effective in enhancing satiety and counteracting that genetic predisposition towards increased appetite.
Interesting. Protein power.
Second, and perhaps even more significantly, physical activity. Regular physical activity appears to strongly attenuate or lessen the effect of the FTO gene on obesity risk. Basically, being active significantly dials down the genetic predisposition.
That's really good news.
It is. And, importantly, studies show that people with the FTO risk allele generally respond just as well as those without it to various weight loss interventions, including diet, exercise, or medication. They might just need to be a bit more mindful or consistent, particularly regarding appetite control and activity levels.
So again, it's predisposition, not destiny. Higher protein for satiety during weight loss and definitely prioritizing physical activity seem key for those with the risk allele.
That's a great summary.
Okay, let's talk about COMT now. This one seems a bit less commonly discussed in general nutrition circles, but sounds important. What does COMT do?
COMT is a really interesting gene. It produces an enzyme called catechol-O-methyltransferase. Its main job is to help break down certain signaling molecules called catecholamines. This includes important neurotransmitters like dopamine, norepinephrine, noradrenaline, and epinephrine adrenaline.
Ah, the stress and motivation neurotransmitters.
Exactly. And it also plays a role in metabolizing catechol estrogens, which are specific types of estrogen metabolites. And importantly for our discussion, the COMT enzyme uses SAME, that universal methyl donor we keep mentioning, to do its job.
So it's linked into that methylation cycle again, like MTHFR and HNMT.
Precisely. It's all interconnected.
Now, what's the key genetic variation or SMP in COMT that gets studied?
The most well-known one is a SNP called Val158met. This variation results in a change in the amino acid sequence of the enzyme. Individuals with the Val version, Valvol genotype, tend to produce a COMT enzyme that's more active and breaks down those catecholamines more quickly, especially at body temperature.
Okay, faster breakdown.
Individuals with the MET version, MET genotype, produce a less active, less stable enzyme, which leads to slower breakdown of catecholamines, particularly dopamine, in certain brain regions like the prefrontal cortex. People with one copy of each, Velmet, fall somewhere in between.
So this difference in enzyme speed, how does that translate into potential traits or tendencies? I've sometimes heard it linked to being a worrier versus a warrior. Is there anything to that?
That's a popular way to frame it, though it's definitely an oversimplification. The idea is that the slower COMT activity in metalleel carriers, warriors, might lead to higher baseline dopamine levels in the prefrontal cortex. This could potentially offer advantages in tasks requiring focus and cognitive stability, but might also make individuals more sensitive to stress and anxiety because the stress neurotransmitters hang around longer.
Okay.
Conversely, the faster COMT activity in allele carriers, warriors, might lead to lower baseline dopamine in that brain region, but potentially better resilience or performance under acute stress as they clear stress catecholamines more quickly. It's also been linked to differences in pain perception.
Fascinating. And what about the connection to estrogen metabolism you mentioned?
Yes, because the COMT enzyme also helps metabolize those catechol-estrogens. Having the slower MET version might lead to less efficient clearance of potentially reactive estrogen metabolites in some individuals. This could theoretically have implications, particularly if other estrogen detoxification pathways aren't optimal.
Okay. And again, COMT relies on methylation, on same. Just to clarify, it doesn't directly break down histamine, right? That's DAO and HNMT.
Correct. No direct enzymatic role in histamine breakdown for COMT. And the provided research didn't link it directly to iron metabolism either. Its main jobs are catecholamines and catechol-estrogens, using same.
Got it. So based on whether someone has the faster VAL or slower MET version, what kind of dietary or lifestyle considerations might be relevant?
Well, for everyone, supporting overall methylation is key, given COMT's reliance on same. So ensuring adequate intake of B vitamins, folate, B12, B6, and magnesium is foundational.
Magnesium seems to come up a lot.
It does. Magnesium can have calming effects on the nervous system, which might be particularly beneficial for metalleel carriers, who might be more prone to stress or anxiety.
Okay.
There's a nuanced view on using supplements like tyrosine, which is a precursor to dopamine. It might be more helpful for VAL carriers to support baseline dopamine, whereas MET carriers might not need it or could even feel worse with too much. It really depends on the individual. For those with slower COMT, metalleel, concerned about estrogen metabolism, supporting phase 2 detoxification pathways could be helpful. This includes things like eating plenty of cruciferous vegetables, broccoli, cauliflower, kale, which support glucuronidation, maybe considering flax seeds for lignans, or potentially supplements like calcium D-glucarate, again, under guidance.
And stress management.
Crucial, especially for metalleel carriers. Practices like mindfulness, meditation, yoga, or simply ensuring adequate downtime could be particularly important. Also, being mindful of things that can overburden the COMT enzyme, like excessive caffeine or alcohol, might be wise for everyone, but perhaps especially for those with slower clearance.
Makes sense. Okay, we're covering some really central genes here. Let's switch gears slightly to HLA-DQ2 and DQ8. These are almost synonymous with celiac disease susceptibility, aren't they? What's their fundamental role?
Yes, they're very strongly linked. HLA molecules in general are part of our immune system, specifically the major histocompatibility complex, or MHC. Their job is essentially to present fragments of proteins, peptides, either from our own cells or from external sources like bacteria or food, to our immune system's T cells.
Like showing the T cells what's going on.
Exactly. They're crucial for immune surveillance. The HLA-DQ molecules are one type, and it turns out that specific versions, namely HLA-DQ2, particularly the DQ2.5 variant, and HLA-DQ8, have a unique structural pocket that happens to bind very strongly to certain peptide fragments derived from gluten.
Gluten, the protein in wheat, barley, and rye. So if you have these specific HLA types, your immune system is basically primed to react strongly to gluten.
In essence, yes. If you have HLA-DQ2.5 or HLA-DQ8, you possess the primary genetic susceptibility factor for developing celiac disease. Over 90% of individuals diagnosed with celiac disease carry HLA-DQ2.5, and most of the remaining percentage carry HLA-DQ8.
Wow, that's a really strong association.
It is. However, and this is a really critical point, these genes are actually quite common in the general population. Depending on the ethnicity, maybe 30–40% of people might carry HLA-DQ2 or DQ8. But only a small fraction of those people, around 1%, actually develop celiac disease.
Ah, okay. So just having the gene doesn't automatically mean you'll get celiac disease.
Absolutely not. It means you have the genetic potential or susceptibility. Other factors are required to trigger the disease. Obviously, exposure to gluten is necessary, but likely other environmental factors, maybe gut microbiome composition, possibly infections, play a role in initiating the autoimmune process in those genetically susceptible individuals.
So, how does the mechanism work in someone who does develop celiac? How do these genes lead to the intestinal damage?
Okay, so when someone with celiac disease and these HLA genes eats gluten, the gluten protein gets partially digested in the gut into smaller peptide fragments. Some of these fragments can cross the intestinal barrier. In the tissue underneath, an enzyme called tissue transglutaminase 2, or TTG2, modifies these gluten peptides through a process called deamidation. It's these deamidated gluten peptides that bind with exceptionally high affinity, really tightly, to the HLA-DQ2 or DQ8 molecules on the surface of specialized immune cells called antigen-presenting cells, APCs.
Right.
When these APCs present the tightly bound gluten peptides to T cells, it triggers a powerful inflammatory immune response specifically directed against gluten. This chronic inflammation in the small intestine leads to damage and flattening of the villi, those tiny finger-like projections essential for nutrient absorption. That's the hallmark of celiac disease.
That makes sense. Such a specific molecular interaction. What about non-celiac weight sensitivity, NCWS? Do HLA-DQ2, DQ8 play a role there too?
The role seems to be much less significant, if any. While some people with NCWS might carry these genes just by chance due to their prevalence, NCWS is generally not considered an autoimmune condition driven by HLA genetics in the same way celiac disease is. The mechanisms behind NCWS are still being researched, but likely involve different pathways, potentially related to other components of wheat like FODPRase or amylase trypsin inhibitors, or perhaps innate immune responses.
Okay, so really distinct from celiac. So for dietary considerations, if someone has HLA-DQ2 or DQ8 and is diagnosed with celiac disease, the treatment is clear, right?
Absolutely. A strict, lifelong gluten-free diet is the only current treatment for celiac disease. Avoiding all wheat, barley, rye, and contaminated oats is essential to allow the intestine to heal and prevent long-term complications.
And for someone with these genes who doesn't have celiac but maybe suspects non-celiac wheat sensitivity?
For NCWS, the approach is usually more about managing gluten intake to a level that avoids triggering symptoms, rather than complete avoidance unless necessary. But first, it's crucial to properly rule out celiac disease through testing before reducing gluten intake, as going gluten-free can interfere with celiac diagnosis.
That brings up testing. How useful is genetic testing for HLA-DQ2-DQ8?
It's very useful, primarily for its high negative predictive value. If someone tests negative for both HLA-DQ2 and HLA-DQ8, it makes the likelihood of them ever developing celiac disease extremely low, essentially ruling it out.
Okay, so it's great for ruling it out.
Exactly. But testing positive only indicates genetic susceptibility. It doesn't diagnose celiac disease on its own. Antibody tests and potentially a biopsy are still needed for a definitive diagnosis if celiac is suspected.
Got it. Very helpful clarification. Okay, we have time to touch on one more crucial set of genes, especially relevant to our previous deep dives. DAO and HNMT. These are the key players in breaking down histamine, right?
Yes, DAO and HNMT are the two main enzymes responsible for histamine degradation in the body. They work in different places and ways.
Tell us about DAO first.
DAO stands for diamine oxidase. Its gene is called AOC1. DAO is primarily produced in the cells lining our small intestine and also in the kidney and placenta. Its main job is to break down histamine that's outside our cells, particularly the histamine we ingest from food in our gut. It prevents excess dietary histamine from being absorbed into the bloodstream.
Okay, the gut guardian against dietary histamine. What about HNMT?
HNMT stands for histamine N-methyltransferase. This enzyme works inside cells throughout the body, including the brain, liver, and airways. It inactivates histamine that's either produced by our own cells, like mast cells, or histamine that has entered the cells from outside.
And HNMT needs something specific to work, right?
Yes, critically, HNMT requires same that universal methyl donor generated through the folate cycle involving MTHFR to transfer a methyl group onto histamine, thereby inactivating it.
Ah, another connection back to methylation. So histamine intolerance, which we've talked about before, happens when there's an imbalance, either too much histamine coming in or the body can't break it down efficiently enough, leading to a buildup in symptoms. Can genetics influence this breakdown capacity?
Absolutely. There are known genetic variations, SNPs, in the AOC1 gene, the gene for DAO, that can lead to reduced production or activity of the DAO enzyme. Several SNPs like RS10156191, RS10497442, RS10497993, and RS205, 212, and 9 have been studied.
And these are linked to symptoms.
Yes, carrying certain combinations of these lower activity DAO variants has been associated in some studies with an increased risk of histamine intolerance symptoms, such as headaches, digestive issues, skin problems, and even potentially contributing to conditions like ADHD, where histamine dysregulation might play a role.
Okay. What about HNMT genetics?
Similarly, there are functional polymorphisms or common variations in the HNMT gene, like C31TT, RS11585W38, that can alter the enzyme stability and activity. These variations have also been linked to conditions where histamine might be involved, like asthma or Parkinson's disease, although the research is complex.
So if someone has SNPs suggesting potentially lower DAO or HNMT activity, are they basically guaranteed to have histamine intolerance?
Not necessarily. Again, it's about predisposition versus certainty. While these genetic variations can definitely increase susceptibility, the actual functional efficiency of these enzymes in real time is influenced by many other factors beyond just the inherited genes.
Such as?
Gut health is a massive one for DAO. Inflammation in the gut, conditions like Crohn's or ulcerative colitis, SIBO, leaky gut, these can all significantly impair DAO production or release regardless of genetics.
Okay, the gut environment is key.
Absolutely. Also, certain medications are known to block DAO activity, things like certain antidepressants, painkillers, and even common medications like acetylcysteine. Alcohol is also a potent DAO blocker. And nutrient status matters too. DAO requires several cofactors to work properly, including vitamin B6, vitamin C, copper, and zinc. Deficiencies in these could impair DAO function. And as we discussed, HNMT's efficiency is directly linked to the availability of Syngyme, which depends on adequate folate, B12, and overall methylation cycle function.
Wow, so it's a real interplay between genetics, gut health, medications, alcohol, and specific nutrient levels.
Oh, exactly. Genetics might set the baseline potential, but these other factors determine how well that potential is realized.
So thinking about personalized nutrition strategies for someone who is experiencing histamine intolerant symptoms and perhaps has a genetic predisposition, what might be involved?
It's usually a multifaceted approach. Starting with a diagnostic low histamine diet for a period is often the first step to see if reducing the external histamine load improves symptoms.
Okay.
Some individuals find significant relief from taking DAO enzyme supplements right before meals containing histamine-rich foods. This helps break down dietary histamine directly in the gut.
Makes sense.
Then, supporting the body's own enzyme function is key. This means ensuring adequate intake of those DAO cofactors, B6, C, copper, zinc, and supporting methylation pathways, folate, B12, maybe choline, to help HNMT function.
And addressing the gut.
Absolutely crucial. Identifying and addressing any underlying gut dysbiosis, inflammation, or permeability issues is often fundamental to long-term improvement in histamine tolerance. Genetic testing for the relevant SNPs and AOC1, HNMT, and even MTHFR can provide valuable clues about potential underlying susceptibilities. And sometimes functional testing looking at stool health, actual DAO levels in blood, or organic acids can add further pieces to the puzzle.
It really highlights the interconnectedness, doesn't it? How MTHFR can impact SAME, which then has implications for both HNMT breaking down histamine and COMT breaking down catecholamines.
Exactly. Nothing works in isolation.
And how these environmental factors, particularly our diet, but also gut health and nutrient status, really determine whether a genetic tendency actually manifests as a noticeable health issue, like ApoE4 and dietary fat, or FTO activity levels.
Precisely. It truly feels like, as you said earlier, our genes might load the gun.
Yeah.
But our environment, broadly defined, is often what pulls the trigger.
That's a very insightful way to put it. These genetic variations seem to be mostly about predisposition, and their actual impact is so highly dependent on the context of our lives, especially our diet.
Yes, which brings us right back to why that one-size-fits-all dietary advice so often falls short. Our individual genetic variations are a massive reason why we all respond so differently to the same foods.
It makes perfect sense when you lay it out like that. Because of our unique genetic blueprints, we just have diverse physiological and metabolic responses. What might be fantastic for one person could be, well, useless or even detrimental for someone else because of their specific SNPs.
Exactly.
Can you give us some more concrete examples? We touched on ApoE4 and saturated fat. What about responses to protein or carbs based on genetics?
Certainly. We mentioned FTO and how higher protein intake might be more beneficial for appetite control during weight loss for those with the risk allele. There are also other genes, like CPS1, where certain variants might affect tolerance to very high protein diets due to potential issues with ammonia detoxification.
Wow, okay.
For carbohydrates, besides APOE4 carriers potentially being more sensitive to refined carbs and sugars, there's the AMY1 gene. This gene codes for salivary amylase, the enzyme that starts starch digestion in the mouth. Humans have varying copy numbers of this gene. Some have few copies, some have many.
And that affects starch digestion.
Yes, generally having more copies is associated with producing more amylase and potentially being better adapted to digest high-starch diets. So individual responses to the amount and type of carbohydrate can definitely have a genetic component. There are also genes related to insulin sensitivity that influence carb tolerance.
So again, it's not that protein or carbs are inherently good or bad across the board, but our genes influence our individual needs and how effectively our bodies process them.
Precisely stated. And this extends beyond just the main macronutrients to how we respond to specific food groups or overall dietary patterns.
Like meat-heavy diets versus plant-based.
Exactly.
Yeah.
For instance, some research suggests interactions between red meat intake and genes like SMAD7 regarding colorectal cancer risk. We mentioned CPS1 and potential issues with ammonia on very high-protein diets. For some people, high meat intake might significantly raise LDL cholesterol or IGF-1 levels depending on their genetics.
Okay. What about plant compounds?
On the plant side, genes like GSTM1 and GSTT1, which are involved in detoxification, can influence how effectively we process beneficial compounds like isothiocyanates from cruciferous vegetables. If you have deletions in these genes, you might process them differently. We already discussed HLA-DQ2-DQ8 and gluten, and there might even be genetic components influencing sensitivity to other plant compounds, sometimes called anti-nutrients like oxalates or lectins. The research there is still evolving.
And ketogenic diets, that's a big one now. Genetics play a role there too.
Oh, definitely. Studies are identifying variants in genes related to fat metabolism like LIPF, GYS2, CETP, and even hormone signaling like GAL, EGTR2 that seem to predict who loses more weight or body fat on a keto diet.
Interesting.
And crucially, lipid responses vary hugely. We talked about APOE4 carriers often seeing LDL spikes. Other genes like LIPC and APOA2 also influence how triglycerides and HDL cholesterol respond to very high fat intake. Some people are hyper-responders. Even cognitive responses to keto might have an APOE component.
Wow. It really sounds like a genetic variation that might be beneficial or neutral in one dietary context could actually be disadvantageous in another. Like maybe good for a low-fat diet, but not a high-fat one, or vice versa.
That's absolutely right. It highlights the context specificity. There's really no single best diet, even for people with the same genotype for one particular gene, because the interplay with other genes and the overall dietary pattern is so complex.
Okay, so our genes influence how we handle macros and specific diets. What about our needs for specific micronutrients beyond folate and gluten, which we've covered?
Yes, genetics can definitely influence our requirements for specific vitamins and minerals too.
What's vitamin D?
Vitamin D is a great example. There are common SNPs in the VDR gene, which codes for the vitamin D receptor. These variations can affect how sensitive your cells are to the vitamin D signal. Someone with a less sensitive receptor might effectively need higher levels of circulating vitamin D to get the same biological effect.
So they might need a higher intake or more sun exposure?
Potentially, yes. And there are also genes involved in vitamin D activation, like CYP2R1 and breakdown, that can influence levels.
What about B12?
Vitamin B12 transport relies heavily on a protein called transcobalamin 2, coded by the TCN2 gene. Certain SNPs and TCN2 can reduce the efficiency of B12 transport into cells, potentially leading to a functional B12 deficiency, even if blood levels look okay on standard tests.
Fascinating. Any others?
Folate transport into cells is influenced by the RFC1 gene. Variations there could increase dietary folate needs. Caffeine metabolism is famously affected by the CYP1A2 gene variations, determine if you're a fast or slow metabolizer, influencing how caffeine affects you and potentially cardiovascular risk with high intake.
Ah, the caffeine gene.
Yep. Vitamin E status can be linked to triglyceride levels, which are influenced by genes like APOA5. Variants associated with higher triglycerides might also correlate with lower vitamin E status, suggesting potentially higher needs. Iron metabolism is heavily genetically influenced by genes like HFE, related to hemochromatosis, TMPRSS6, and TF.
And maybe antioxidant needs.
Yes, genes coding for key antioxidant enzymes like catalase, CAT, superoxide dismutase, SOD2, and glutathione peroxidase, GPX1, have common variants that can reduce enzyme efficacy. This might mean individuals with less efficient versions have a higher requirement for dietary antioxidants to manage oxidative stress.
Inflammation, too.
Definitely. Variants in genes coding for inflammatory cytokines like interleukin-6, IL-6, or tumor necrosis factor, TNF, can predispose individuals to heightened inflammatory responses, making anti-inflammatory dietary strategies potentially even more crucial for them.
It just keeps reinforcing this central theme, doesn't it? Genetic individuality really trumps universal dietary roles,
Yeah.
and it helps validate those personal experiences, those intuitions people have about needing more of something or being sensitive to something else.
Exactly.
Food truly is this incredibly complex form of biochemical information that interacts uniquely with each of our biological systems.
Beautifully put. And to really grasp the context for some of these predispositions and why modern diets can be problematic for many, it's helpful to look back at our evolutionary history.
Right, the whole gene-diet mismatch idea, the theory that our genes haven't caught up with our modern food environment.
Precisely.
Yeah.
Our genetic makeup was sculpted over hundreds of thousands, even millions of years, by the diets our hunter-gatherer ancestors consumed, predominantly whole, unprocessed foods, wild plants, and animals.
Yeah.
Over these vast timescales, genetic adaptations arose that helped us thrive on those types of diets. Think about this shift from primarily plant-based diets to including more meat, the advent of cooking, which changed nutrient availability. These things drove genetic selection.
But then things changed relatively recently in evolutionary terms.
Dramatically so. First, the agricultural revolution, around 10,000 years ago, introduced grains and dairy on a large scale. This led to some adaptations, like lactase persistence, the ability to digest milk sugar into adulthood, evolving independently in different populations that relied on dairy, or variations in the AMY1 gene for starch digestion we mentioned earlier.
Right, but the really big shift was much more recent.
Yes, the Industrial Revolution, and especially the last 1,500 years, have brought unprecedented changes to our food supply at a speed that genetic adaptation simply cannot keep pace with.
So this mismatch, what are the key features of the modern food landscape that create this clash with our older genetic programming?
Several things. The massive influx of ultra-processed foods, loaded with refined sugars, unhealthy fats, salt, and novel additives, drastically altered macronutrient ratios compared to ancestral diets, a significant reduction in the sheer diversity of plant foods and therefore phytonutrients we consume, lower fiber intake. These are huge shifts.
And the implications for modern chronic diseases.
The gene-diet mismatch theory posits that this dissonance is a major driving force behind the epidemics of obesity, type 2 diabetes, cardiovascular disease, and potentially even some autoimmune conditions and chronic inflammatory disorders.
How so?
Our genes might still be geared towards efficiently storing energy from intermittent feasts, which becomes a major liability in an environment of constant, readily available, high-calorie food leading to obesity. Our glucose regulation systems might be overloaded by constant high-carb, high-sugar intake, contributing to insulin resistance and type 2 diabetes. Our cardiovascular systems might suffer from the altered fat profiles, high sodium, and low intake of protective phytonutrients.
It makes intuitive sense. So things like the paleo diet are essentially an attempt to address this mismatch, to eat more in line with our presumed ancestral patterns.
Yes, that's the core idea behind the paleo diet, mimicking the food groups thought to be available to Paleolithic humans. It emphasizes whole foods like meat, fish, vegetables, fruits, nuts, and seeds, while excluding grains, legumes, dairy, and processed foods.
Does it work? Are there concerns?
It can offer benefits for some people, like improved weight management, blood sugar control, and lipid profiles, likely due to its focus on whole foods and exclusion of processed items. However, concerns include potential nutrient deficiencies, if not well planned, like calcium or fiber, the cost and sustainability, and the relative lack of long-term, large-scale clinical trial data compared to other patterns like the Mediterranean diet.
So maybe not a perfect replica or solution, but the underlying principle of being mindful of our evolutionary legacy.
Yes.
And prioritizing whole, minimally processed foods probably holds a lot of value for most people, regardless of whether they follow a strict paleo template.
I think that's a very reasonable takeaway. And we absolutely cannot have this conversation without considering another critical player that sits right at the intersection of our genes and our diet, the gut microbiome.
Yes. The trillions of microbes living in our gut, they're like a whole other organ, aren't they? And they must interact with both our genes and what we eat.
They absolutely do. It's a three-way conversation. And interestingly, our own host genetics actually play a role in shaping the composition of our gut microbiome from the start.
Really? How?
Studies, especially twin studies, have shown that the abundance of certain types of microbes in our gut is partly heritable, meaning it runs in families to some extent. Specific bacterial families like Christensen-Lachey show strong heritability.
Wow.
Scientists are identifying specific human genes, particularly those involved in immunity, like NOD2 or FUT2, which influences secretions in the gut, and metabolism, that seem to influence which microbes are more likely to colonize and thrive in our individual gut environments. Our genes can affect things like gut mucus production or bile acid profiles, creating a selective habitat.
So our genes help set the stage for the microbiome.
Exactly. And then, in this incredible feedback loop, the gut microbes that take up residence can, in turn, profoundly influence our host gene expression and even our epigenetics,
How do you do that?
primarily through the metabolites they produce. When our gut bacteria ferment dietary fiber, for instance, they produce short-chain fatty acids, like butyrate. Butyrate is not only fuel for our colon cells, but it's also a known HDAC inhibitor. Remember those histone modifications?
Right, so microbial products can directly influence our epigenetic controls.
Precisely. Butyrate can help keep certain beneficial genes active. Other microbial metabolites, like secondary bile acids or even precursors to neurotransmitters, also act as signaling molecules that can communicate with our host cells and influence gene activity related to metabolism, immunity, and even brain function. It's a constant chemical dialogue.
So the food we eat feeds the microbes. The microbes produce compounds, and those compounds talk back to our genes. It really brings that whole food as information concept full circle, with the microbiome acting as this crucial translator.
That's a perfect description. And beyond gene expression, the microbiome also directly impacts how we metabolize nutrients. They help us extract energy from indigestible fibers. They synthesize certain vitamins, like some B vitamins and vitamin K. They transform bile acids, and they even help metabolize drugs and dietary compounds like polyphenols, sometimes converting them into more active forms.
So they're essential metabolic partners.
Absolutely. It really underscores this triad. Diet is the input, our host genetics provide the background susceptibility and influence the environment, and the microbiome acts as a dynamic mediator and effector, translating dietary signals and influencing host responses. Disruptions in this intricate diet-gene microbiome balance are increasingly implicated in the development of many chronic diseases. It relates back to that idea of the terrain we discussed previously.
It's such a complex, interconnected system, which leads us naturally to the idea of personalized nutrition. If everything is so interconnected and individual, how can we use this knowledge practically?
That's the goal. Personalized nutrition aims to move beyond generic advice and provide dietary recommendations tailored to an individual's unique biological makeup, considering their genetics, microbiome, metabolism, lifestyle, and health goals.
So what are some of the practical ways this is starting to be applied or could be in the future?
While we can offer more targeted dietary advice, we've discussed examples like specific fat recommendations for APOE4 carriers, or maybe emphasizing higher protein for someone with the FTO risk allele trying to manage weight, or suggesting increased omega-3 intake for someone with genetic variants predisposing them to higher inflammation.
Right.
It also opens the door for more proactive strategies for disease prevention. If someone knows they have a significantly increased genetic risk for, say, type 2 diabetes or celiac disease, they can implement targeted dietary and lifestyle interventions earlier and more consistently.
Optimizing nutrient status, too.
Definitely. By understanding genetic variations that might affect the absorption, transport, or metabolism of specific nutrients like vitamin D, B12, folate, iron, vitamin E, we can help individuals ensure they're meeting their unique requirements, which might differ from the standard RDAs. It helps move beyond guesswork. Ideally, this involves integrating omics, data genomics, epigenomics, metabolomics, microbiome analysis for a truly holistic picture.
Can you give us a few more specific examples? Let's say for histamine intolerance, how might a personalized approach look?
Okay, for histamine intolerance, genetic testing might reveal SNPs in DAO, HNMT, or MTHFR, indicating potential bottlenecks. This could reinforce the trial of a low histamine diet. Based on genetics and symptoms, DAO enzyme supplementation might be considered. We'd ensure adequate intake of cofactors like B6, C, copper, zinc. If MTHFR variants are present, methylation support, active Bs, choline, would be emphasized. And critically, investigating and addressing underlying gut health issues via functional testing, stool analysis, DAO levels, organic acids, would be paramount.
Very comprehensive. What about metabolic health, like obesity or type 2 diabetes?
Here, knowing FTO genotype might guide protein recommendations for satiety. APOE genotype could influence the balance of fats and carbohydrates suggested. Some emerging genetic markers might even help predict who is likely to respond better to a ketogenic versus a lower-fat diet for weight loss or blood sugar control. It helps tailor the macronutrient approach.
Cardiovascular health.
APOE genotype is key for tailoring dietary fat advice, saturated versus unsaturated. MTHFR status informs the importance of monitoring homocysteine and ensuring adequate B vitamin intake, folate, B12, B6. Other genes influencing lipid metabolism, like those affecting LDL particle size or triglyceride response, could further refine recommendations.
And cognitive health, especially with the APOE4 concerns.
For APOE4 carriers, emphasizing brain protective nutrients like omega-3s, DHA, antioxidants, and choline becomes even more critical. Dietary patterns like MIND or Mediterranean are often recommended. COMT genotype might offer nuances, maybe ensuring adequate tyrosine for vulval warriors, or focusing more on stress management and estrogen balance support, cruciferous veg for MET warriors, while ensuring magnesium intake.
It sounds like genetic testing can be a really valuable tool in building these personalized strategies. What do you see as the main potential benefits, but also the limitations or challenges people should be aware of?
The potential benefits are significant. Genetic information can offer insights into why someone might respond a certain way to certain foods, helping explain past experiences. It can motivate positive lifestyle changes by highlighting predispositions. It empowers individuals with knowledge about their unique biology and allows for more proactive health management. Understanding the mechanism can be very powerful.
But it's not a crystal ball, right?
Absolutely not. That's where the limitations come in. The interactions between genes, diet, lifestyle, and the microbiome are incredibly complex, and our scientific understanding is still evolving rapidly. A single gene rarely tells the whole story.
Right.
Interpretation requires expertise. Direct-to-consumer, DTC, genetic tests can provide interesting data, but the interpretation offered is often simplistic and can be misleading or cause unnecessary anxiety without professional context. There are ethical considerations, too, around data, privacy, and potential discrimination. Cost and accessibility can also be barriers. And crucially, genetics indicate risk or predisposition, not certainty or destiny.
So how should we frame personalized nutrition so it feels empowering and accessible rather than deterministic or scary?
I think the focus needs to be squarely on empowerment and agency. It's about using this information as a tool to make more informed, actionable choices, primarily centered around whole, nutrient-dense foods and positive lifestyle habits. It's about optimizing health with the genes you have, not feeling limited by them.
Yeah.
It can help reduce the frustration people feel when generic advice fails them. It provides a why behind certain recommendations. But the core message should be positive. Understanding your biology gives you more levers to pull for better health. Education and making guidance more accessible are key to realizing this potential.
Looking ahead now, what role do you see technology, maybe like AI, playing in advancing personalized nutrition?
Oh, I think AI and machine learning are going to be absolutely crucial. The sheer volume and complexity of data involve genomics, epigenomics, transcriptomics, proteomics, metabolomics, microbiome data, plus detailed diet and lifestyle tracking. It's beyond human capacity to integrate effectively.
Right.
AI algorithms can analyze these vast data sets to identify complex patterns, predict individual responses to specific foods or diets with much greater accuracy, and ultimately help generate highly personalized, dynamic recommendations.
So AI could help create the actual advice.
Yes, potentially powering personalized guidance tools or apps. It can also massively accelerate research by identifying new gene-diet interactions or biomarkers. AI is really key to bridging that gap between the overwhelming complexity of the data and providing clear, actionable advice for individuals.
It sounds like we're really on the cusp of some potentially revolutionary changes in how we approach food, nutrition, and health.
I truly believe we are. It's a paradigm shift towards a much more nuanced, individualized, and ultimately more effective understanding of what it means to truly nourish ourselves based on our unique biology.
So to bring this incredible deep dive to a close, what's the single most empowering message you want our listeners to take away from this whole discussion on nutrigenomics?
I think the most empowering message is this. While your genes absolutely provide an important blueprint, they are not your destiny. Your diet and lifestyle choices offer a significant degree of control over your health outcomes. This happens through that intricate interplay of gene-diet interactions, through the dynamic nature of epigenetics, and through influencing that incredible ecosystem within you, your microbiome. Remember, food is powerful biological information that interacts uniquely with you.
That's a fantastic summary. And for listeners who have been captivated by this and want to explore it further for themselves, what would be your practical advice?
First, I'd encourage you simply to become more mindful and curious about your own individual responses to different foods and dietary patterns. Pay attention to how you feel. Second, If you want to learn more, seek out reliable information from reputable scientific sources, perhaps starting with some of the concepts we discussed today. And if you're considering genetic testing or making significant dietary changes based on this, please consult with qualified healthcare professionals, doctors, registered dietitians, or nutritional therapists who have specific expertise and training in nutrigenomics and functional medicine.
Be cautious with just the raw data from DTC tests.
Yes, definitely be cautious about trying to interpret complex genetic data or making drastic changes based solely on DTC reports without professional guidance. Remember, the bigger picture involves your whole health history, lifestyle, and other factors.
Excellent practical advice. And that brings us, as always, to our final provocative thought for you, our listener, to mull over. Considering this incredibly intricate, dynamic interplay of your unique genes, your daily diet, and your gut microbiome, what might truly eating in tune with your biology actually look like for you in the context of our modern food world?
Something to think about.
Definitely. Thank you so much for guiding us through this complex and fascinating topic.
My pleasure. It was great discussing it.
And thank you, our listeners, for taking this deep dive with us.